will have GC GC sequence, and it will form a GC hairpin. This hairpin formation is necessary because it will stabilize the newly synthesized messenger RNA, because if this strand is straight, just straight, it will be unstable and it will be highly reactive, and ultimately it will react with something in the cytoplasm and it will be destroyed. So in order to stop, there is rho factor. When rho factor is reached, the messenger RNA which is formed, it takes the shape of this GC hairpin, and at the terminals of this hairpin there is a uracil, 4 or more uracil ribonucleotides. This hairpin causes RNA polymerase to stop the synthesis. Now when this hairpin is formed, it will lock the newly synthesized messenger RNA, and the messenger RNA will become stable, and ultimately the whole process of transcription will stop. Now when it comes to bacteria, since they have no proper nuclear membrane, so the messenger RNA, when it will be synthesized, it will be removed from here and it will directly enter into the cytoplasm. On the other hand, in prokaryotes there is proper nucleus, so the messenger RNA has to cover a long distance from inside of the nucleus to the ribosomes outside in the cytoplasm. The eukaryotic messenger RNA is modified in several ways. It is first modified and protected so that it can safely complete its journey from nucleus to cytoplasm. For example, what happens is that messenger RNA will be provided with a tail and a head region or the cap region. It will be covered with a cap and a tail. The cap is in the form of 7-methyl GTP. Cap is 7-methyl GTP, which is linked at the 5 end, 5', and tail is in the form of poly A. Tail is poly A tail, which is attached at the 3' end. Now what is the function of this cap and tail? The cap and tail, they will protect the newly synthesized messenger RNA so that it can safely travel from this DNA, from the nuclear atmosphere into the cytoplasm. So it will safely move from nucleus into the cytoplasm, and ultimately it is protected, and no enzymes like nucleases and phosphatases which are present along this whole distance and along this whole journey, they will not be able to damage our newly synthesized messenger RNA. Now for your convenience, I am going to repeat and revise the process. First of all, this is the initiation step. The process is initiated when a factor, sigma factor, which is actually a part of this RNA polymerase, is attached to this promoter site. Promoter site is a site present before our desired gene of interest, and is this sequence, when the sigma factor will bind to this sequence, it will initiate the whole process correctly. Then the next process is elongation. Elongation will be carried out according to the sequence of template strand, because template strand is contrary to the coding strand, it is complementary to the coding strand. So if we want to copy this strand, we will use the template strand. This is coding strand because its code is to be copied. For example, this code says that you have to synthesize insulin. So what will happen, that we will not use this code, we will use the complementary code, and the complementary code will be in 3 to 5 direction, which will guide the 5 to 3 direction formation of the new messenger RNA. At the end, this is termination. Another factor from RNA polymerase, which is known as the Rho factor, when it will be attached to this ending sequence, end sequence, the whole process will be terminated. And as soon as the process is terminated, the newly synthesized messenger RNA will take the shape of GC hairpin with GC base pairs, which are then followed by AT base pairs. And at the end, there will be 4 or more uracil ribonucleotides. This hairpin will stabilize the newly synthesized messenger RNA, otherwise it will be very unstable and very reactive. In case of bacteria, the newly synthesized messenger RNA will directly be in cytoplasm, whereas in eukaryote it has to cover a long distance from nucleus into the cytoplasm. So it is modified in variety of ways. It is protected by a cap and a tail. The cap is 7-methyl GTP and the tail is the poly A tail. They will protect by the newly synthesized messenger RNA from the action of various enzymes like nucleases and phosphatases. So this is all about the process of transcription. I hope it is very much clear to you. In the next video, we will be discussing the genetic code. Till then, keep studying, keep watching office.com.pk. Thank you so much for your time, and please do not forget to subscribe to our channel. Thank you so much. Allah Hafiz.